Hey guys, welcome to Rocky by Chem series. Today's tutorial is about complementary DNA library construction. If you enjoy the content, please support the channel by hitting the subscribe button and then giving this video a like. Thank you so much as you are doing so. In my previous video on DNA library construction, I explained that DNA libraries can be grouped into genomic DNA library and complementary DNA library. Genomic DNA libraries are ideal for prokaryotic organisms, and this is because they have relatively smaller genome size and then they lack intronic sequences as well. On the other hand, complementary DNA libraries works best for eukaryotic organisms, and this is because eukaryotic organisms have larger genome size and then they also have introns in their genome. For us to appreciate what we are going to do today, I would like us to do a very quick recap about how gene expression occurs in eukaryotic organisms. So during gene expression, a segment of a gene undergoes transcription to produce an mRNA molecule, and usually the mRNA undergoes translation to produce proteins. Now, when we get to produce these proteins, then the proteins produce whatever function that it has to play or like it has to perform in the body. But there is this issue about eukaryotic gene expression. In eukaryotic organisms, gene expression is tissue specific and then spatial temporal. When I say gene expression is tissue specific and then spatial temporal, what I'm trying to mean is that although every single cell in the body possess the same chunk of genomic DNA. When you pick any cell in the body, there's genomic DNA is the same in all the cells. However, the expression of certain genes differs from cell to cell or tissue to tissue. That means the genes that are expressed in a cell like maybe the genes that are expressed in maybe the eyes may not be the same as the genes that are expressed in the pancreas. In other words, the genes that are expressed in the muscles will not be the same as the genes that are expressed in any other parts of the body. And aside that, the expression also occurs at different stages during development. So this describes the spatiotemporal and then tissue-specific nature of gene expression in eukaryotic organisms. So due to this, in case you want to study a particular gene of interest, you have to target the tissue or the cell in which the gene of interest is expressing. And then you extract mRNA from that cell and then you use it for any downstream application that you want to study. So for instance, if you are interested in studying a gene like the gene that codes for insulin, you have to go straight to the pancreas and then isolate your mRNA from the pancreas. Now, the problem with mRNA is that in case you want to use mRNA for any downstream applications, mRNA itself is an unstable molecule. It is highly unstable and then it quickly degrades. So you have to convert the mRNA back to cDNA through a process called reverse transcription. And the enzyme responsible for this work is called reverse transcriptase. We need to convert the mRNA, and the reason is that mRNA in itself is what? An unstable molecule that quickly undergoes degradation. So let's delve deep into how the cDNA is obtained and then how the cDNA is used for the library construction. That is the real business for the day. So before then, let's get familiar with the meaning of cDNA library. I think for my previous video on DNA library, I defined what a DNA library is. For a cDNA library, it is a collection of clones containing cDNA that is synthesized from the mRNA of cells where the gene of interest is expressing or the gene of interest is expressed. The main emphasis is on the cells where the gene of interest is expressed. That is why when I started, I said that you have to target the cell in which your gene of interest is expressed, and then you extract mRNA from that particular cell before you can obtain a complementary DNA form of the gene that you are interested in studying. Now, let's talk about the workflow of cDNA library construction, I would like to take you through the workflow before delving deep into the detail of each of the various steps. So the first step involves mRNA isolation from the cell or the tissue of interest, and this is followed by synthesis of cDNA using the mRNA as a template, and then moving on to perform cloning. That is, we will ligate our isolated cDNA with an appropriate vector to obtain a clone, and then we will transform bacteria with the clones that we have generated. So I'm going to pick each of the steps one after the other and then talk about it in detail. Now, let me start with the first step, that is mRNA isolation and then purification. So, we have to target the cell in which the gene of interest is expressing. So, for instance, you want to do extensive studies of the insulin gene. You have to look no further than going directly to the pancreas and then extract mRNA from the pancreatic beta cells. And then, after extracting your mRNA from your target cell, then we can go ahead and then purify the mRNA. So the mRNA isolation step may involve the use of a number of reagents that will break the cell membrane as well as break the nuclear membrane for us to access the nucleic for us to access the nuclear material, that is both the DNA and then the mRNA, and then we will degrade the DNA portion by treating it with an enzyme called DNase. Now that we have obtained this mRNA, we need to purify it before using it for any downstream application. So the purification of the mRNA involves passing it through a column that is packed with an oligo DT molecules. So the oligo DT is just a short stretch of DNA sequences that are made up of only T nucleotides. The idea here is that when you pass the mRNA through this column, the poly-A tail of the mRNA would anneal to the oligo-DT that is packed in the column. And by increasing the concentration of salts in the column, then the purified mRNA having the oligo-DT annealed to the poly-A tail would be obtained. So we can then use this purified form of the mRNA with the oligo-DT nucleotides attached to the poly-A tail as our template for the synthesis of our cDNA. So this ends the mRNA isolation and then the purification. So let's move on to the next step, that is cDNA synthesis. The synthesis of cDNA is divided into two. We have first strand synthesis and then second strand synthesis. So we aim at getting double stranded cDNA. So we have to first of all synthesize our first strand and then move on to synthesize our second strand. So the first strand synthesis involves the use of the isolated mRNA as a template, and in the presence of an enzyme called reverse transcriptase, then the mRNA will be used as a template for the synthesis of the first cDNA strand. And at the end of the first strand synthesis, we would obtain a hybrid of mRNA and then a cDNA. So this is a cDNA mRNA hybrid. That is the product from the first strand synthesis. And after obtaining this hybrid, this is not what we are aiming or we are aiming at obtaining. We want to get a double-stranded cDNA. So we have to degrade the mRNA portion, leaving the cDNA. And that is where the second strand synthesis starts from. So for the second strand synthesis, we will treat the cDNA mRNA hybrid with an enzyme called ribonuclease or an alkali. Any of them, you can either use an alkali or a ribonuclease, and any of them will degrade the mRNA portion, leaving a single-stranded cDNA. And this single-stranded cDNA would serve as a template for the synthesis of the second strand. And this work is done by an enzyme called DNA polymerase 1. And at the end of the second strand synthesis, we are going to generate a double-stranded cDNA molecule. This is what we aim at obtaining. But the problem is that this double-stranded cDNA that we are having is having a hairpin loop at the end, we don't like the hairpin loop. We don't want the hairpin loop. So we would have to treat it with an enzyme called S1 nuclease. And the S1 nuclease would trim off the hairpin loops attached to one end, leading to a double-stranded cDNA molecule with no hairpin loop. Now we can just move on and then use this double-stranded cDNA molecule for our library construction. And before we start or before we continue with the library construction, we need to create our clones. And then the clones creation involves ligation of our insert, which is the cDNA molecule, with a library with a vector that is a plasmid or any other form of vector like bacteria artificial chromosome or maybe yak vector and then etc. Now, let's look at our cDNA molecule that we are going to ligate with the vector. So, looking at this molecule, it is having a blunt end. When we say a DNA molecule is having a blunt end, it means that the length of both strands are equal. Both strands are of the same length. And due to that, when we try to seal it with another fragment of DNA from the vector, it becomes so difficult. So we try to generate what we call a sticky end. And the sticky end generation involves degrading the length of one strand and then leaving the other so that one strand would be longer than the other. That is what a sticky end is. So when you are ligating as two sticky ended DNA molecules, it becomes so easy. But the problem is that when we try to treat our cDNA directly with a restoration enzyme, some desirable sequences might be lost. So we attach a short fragment of DNA called linkers to the ends of the cDNA molecule before we carry out the restoration digestion. So these linkers that we have attached to the ends of the cDNA molecules possess restoration sites within them. A restoration site is just a particular stretch of sequences that is recognized by a specific restoration enzyme, and that enzyme will recognize that region alone and then cut in a specific manner. So by treating this with a restoration enzyme, the restoration enzymes will cut at the linker regions to generate a sticky ended insert or a sticky ended cDNA molecule as our insert. Now, still on the ligation, let's move on to start with our plasmid digestion or our vector digestion. So we would then move on to treat the vector, that is the plasmid, with the same restoration enzyme that we used earlier. The same restoration enzyme that we used earlier has to be treated or has to be used to treat the vector once again. And by treating the vector, it would also digest it at specific regions to create an open form or a digested form of our vector. And we can then combine these two. We can combine the digested plasmid and then the digested cDNA insert to obtain our clone. And we can then move on to use this clone for the transformation. Now, on the fourth step involves transformation of the bacteria with clones. We have to select competent cells and then use it for the transformation. A competent cell is a cell that is just that is easy to take up any plasmid. So we in the lab we use some group of bacteria, some group of E. coli strains called the Nova Blue E. coli. We also have the DH5 alpha cells. They are all competent cells that can be used for transformation in the lab. So the idea behind transformation is that we want to open the bacteria cells and then insert the clones into them. And after that, we seal it. And after sealing it, then the bacteria will replicate the clones into multiple copies. That is the idea behind transformation. So this is our clone, and then we will insert the clone into the bacteria, and that is the transformation. And after doing so, the bacteria will replicate through cell the bacteria will undergo replication and then cell division to obtain multiple clones containing multiple I mean containing to produce multiple clones. So this is what we have to know about the cDNA library construction.